Okay, everyone, here we are back with part two of the online lecture on Chapter 10, Photosynthesis. So we had finished up at the end of the last lecture talking about the spectrophotometer and the way that it measures which wavelengths of light are absorbed by chlorophyll and that sort of thing, and other pigments. Next thing to talk about is what we call an absorption spectrum, okay? So an absorption spectrum basically is a figure that tells us or shows us what wavelengths of light a pigment, like chlorophyll for example, absorbs and which ones it doesn't absorb. There's also another thing we can look at called an action spectrum, which we'll see here in a minute. But what I want to talk about first of all is an experiment that was conducted in the 1880s by a German biologist named Theodor Engelmann. All right, so Engelmann was interested in understanding what colors of the visible spectrum, what wavelengths of the visible spectrum were responsible for running photosynthesis. And so he conducted a very simple but very elegant experiment, which you basically see here in this bottom panel. So let's talk about that first. What Engelmann did is he took a species of filamentous algae, that's what you see right here, and he grew it in a culture medium, a culture dish, with all the nutrients and things it would need to survive. Now, this algae, he reasoned, when it was undergoing or undertaking photosynthesis, would produce oxygen as a byproduct, as plants do. So he therefore included in the culture dish or culture medium a species of aerobic bacteria. So aerobic meaning those bacteria require oxygen. All right. Then he's shown the visible colors of the spectrum, the rainbow, as you see here, from 380 to 750 or so nanometers across that filament of algae. He took light and split it out with a prism. And then he looked to see where the bacteria grew best. And he did bacterial counts after some time. What he found was that he had a very high population or high growth rate of bacteria in the purple, blue, violet region, nothing really in the green, little in the yellow, and a second peak in the orange and red. So the bacteria here were doing very well, bacteria here were doing very well, and remember, these are bacteria that require oxygen. So based on that, Engelmann surmised that green light is not actually being used by the algae in this case because there was no oxygen being produced there, as evidenced by the fact there were no bacteria growing there. But this section of algae and this section of algae, where you had the peaks in bacterial growth, were producing lots of oxygen to support that bacterial growth, so it must have been the orange and red and the blue and violet and purple that were running photosynthesis. Okay, so in our next figure above here, we see what's called an action spectrum. This is showing us the rate of photosynthesis measured by the amount of O2 produced. So what this is saying is that at 450 nanometers, for example, we have a peak of O2 production. We have almost nothing in the green, a second peak around 660, 670, something like that. And if you follow that curve, right, it matches very precisely the bacterial population curve or growth curve. Those two are saying the very same things. They're saying that it's the purple and blue wavelengths and the orange and red wavelengths that run photosynthesis. The green wavelengths do not. So why is this? Well, to understand that, let's look at the absorption spectrum that you see here. What this is showing you is three different pigments. You see chlorophyll A, which is the primary photosynthetic pigment, another pigment called chlorophyll B, that's an accessory pigment, and also another type of accessory pigment called carotenoids. Well, notice that chlorophyll A and chlorophyll B have peaks at slightly different wavelengths on the blue-violet end, and two more peaks at slightly different wavelengths on the orange-red end. So by having two different types of chlorophyll, the plant can actually absorb over a broader range of the wavelength of the spectrum, and that translates into more energy being captured and more sugar being produced. Notice that chlorophyll A and B do nothing in the green and really in the yellow either. Okay. Now, what do the carotenoids do? The carotenoids are an accessory pigment They do absorb wavelengths of light, but for the most part, it's not being funneled into photosynthesis. What they do is they act as a sun shield to protect chlorophyll A and B. If you take chlorophyll A and B and extract it from a leaf, you extract it from all the machinery of the leaf cells and put it in a vial and expose that to light, eventually that chlorophyll A and B break down. It turns into this brown, muddy-looking substance. So what the carotenoids do in the leaf cells along with the chlorophyll A and B is they act as a sun shield and protect chlorophyll A and B from that excess radiation or sunlight, which would cause chlorophyll A and B to break down. All right, that's what they do. But if you look overall at the wavelengths of light, there's a large peak here, a trough there, and a second peak here. Matches up very nicely with the action spectrum and also, again, with the population growth curve of the bacteria in Engelmann's experiment. Okay, so based on this experiment, Engelmann concluded that that plants running photosynthesis do not use green light. They use purple, blue, violet, and orange and red. Okay, so let's take a look at the chlorophyll molecule. Here you see both chlorophyll A and chlorophyll B. We'll get to the difference in a minute. On this end, what we have is the hydrocarbon tail. This is what actually anchors the molecule into the phospholipid bilayer membrane of the thylakoids, the little dinner plates in the stack that I mentioned inside the chloroplast. So that anchors it in the membrane. And then up here on this end, we have what's called the porphyrin ring. This is the light-absorbing portion of the molecule. There's a magnesium atom there at the center, and when chlorophyll is exposed to light in the absence of carotenoid pigments that would protect it, that magnesium atom pops out and the whole thing breaks down. Okay, so chlorophyll A has a methyl group right there, CH3, you see it here. Chlorophyll B at this location has a CHO group. That is the only difference between A and B, and that one difference is what causes them to absorb at different wavelengths. All right. So now let's start to talk about what it is that happens to electrons and chlorophyll when chlorophyll is exposed to light. Okay, so we refer to this process as excitation. So what that means from like a biochemical standpoint is that there's energy being applied to a molecule or electrons or whatever. Way back at the beginning of the class, in January, I talked about, I think it was in chapter 2, how electrons around atoms can move further away from the nucleus of the atom when the atom absorbs energy and move closer to it when the atom gives off energy. And I said then, this is the whole basis of how photosynthesis works that we would see in chapter 10. And well, here we are now in chapter 10. So if you take atoms and expose them to light, the electrons around those atoms will bump out to higher energy levels or higher electron levels, further away from the nucleus. They can then fall back down and give that energy that they absorbed back off as different forms of energy, very often heat or different wavelengths of light than the light that was absorbed. This is exactly what electrons do. So, for example, let's say you take a vial of chlorophyll that you see here. You extract the chlorophyll from a leaf, and it doesn't look red like that. It's this beautiful emerald green color. But you shine light on it, and it turns this sort of blood red color. It's kind of an eerie effect if you've ever seen it. What's going on there is something called fluorescence. Okay, so here you have a chlorophyll molecule. Photon of light comes in, is absorbed, and the electrons around the atoms in the chlorophyll get bumped up to a higher energy level. That's called the excited state. All right. Now we're going to see later on there's a way to trap those electrons in that excited state. But in the chlorophyll that's been extracted from the cell, that's no longer an option. The electrons fall back down to their ground state, and they give off the energy they absorbed as heat and light. The light they give off is now in the red wavelengths, and that's why this appears to be red. That vial of chlorophyll will also start to heat up. If you hold it in your hand, you can feel it starting to heat up. So fluorescence is showing you what it is electrons are doing that actually allows photosynthesis to do what it does. Okay. All right, so now we're going to start talking about the machinery of machinery in the cell or in the chloroplast that's involved in running photosynthesis. We have machines basically called photosystems. Okay, and in photosynthesis we have two of them. So what is a photosystem? Well, it consists of what's called a reaction center complex. This is a series of proteins, and those are surrounded by what are called light harvesting complexes. And the light harvesting complexes contain the chlorophyll molecules. Okay, so this is essentially what we're looking at right here on the left. Okay, so the whole thing that I'm circling here with the pointer, that is the photosystem. In the middle, you have the reaction center complex. It's a series of proteins and a special pair of chlorophyll molecules that we'll get back to later. On the outside, you have the light harvesting complex. This is where most of the chlorophyll molecules, shown in green, are found. So what's happening here is that a photon of light comes in. So now realize we're talking about light as a particle of light, a photon, a discrete packet of light energy, comes in, it impacts or is absorbed by a chlorophyll molecule. The electrons in that chlorophyll molecule get bumped up to a high energy state, and they immediately fall back down. The energy then is transferred to the next chlorophyll. Its electrons get bumped up, they fall back down, the energy is transferred to the next one, and so on down the line, until eventually the energy is donated to a special pair of chlorophyll molecules right here. It's a special pair that actually lose their electrons. Their electrons get bumped up, and now there's a protein called a primary electron acceptor that grabs onto those electrons. So think of it this way. Imagine you're shooting hoops, playing basketball, and you shoot the ball up to the goal, and it gets wedged between the rim and the backboard. It gets stuck there. When you shoot it up, it's going from low potential to high potential. Okay, you're injecting energy into it, as we talked about in Chapter 8. But then because it gets stuck there, wedged between the rim and the backboard, it remains at high potential energy. If it falls back down, it gives its energy back off again. So these electrons get stuck, or wedged, so to speak, in that primary electron acceptor protein, and that traps the energy of sunlight in them. All right, we'll see later on what happens with that energy, how it's actually used to build ATP molecules a little bit later on. Okay, on the right-hand side over here, you see a computer-generated diagram of a photosystem. It's not really that helpful. It shows you it's complex, but this is how you really want to think about it over here. All right. Okay, so we've talked about that primary electron acceptor. If you remember in Chapter 9 on aerobic respiration, I talked about oxygen acting as the terminal electron acceptor, the final electron acceptor, pulling those electrons down that transport chain. So the electrons that have been energized by photosynthesis and then get pulled out of the sugar molecule by cell respiration ultimately end up back in water. Well then, water is split at the beginning of photosynthesis, and the electrons come right back out again. They get energized by these photosystems that we're talking about here, and then they get packaged into a sugar molecule. So it's one large cycle with essentially photosynthesis energizing electrons and cell respiration pulling those energized electrons out of food, siphoning off their energy and using that to make ATP, and putting them back in water again, where photosynthesis then pulls them back out and re-energizes them. It's all one big cycle, okay? So this is solar-powered transfer of an electron from a chlorophyll molecule to the primary electron acceptor. That is the first step of photosynthesis. It's the first step of the light reactions, okay? All right. So we basically have two photosystems. Sorry guys, my phone's ringing. We basically have two photosystems, okay? We have what's called photosystem 1 and photosystem 2. So we just call them PS1 and, PS2. All right, now here's the thing. They're in a line, they work in a sequence, and it turns out that photosystem 2 actually happens first, and photosystem 1 happens second. This is a confusing aspect of the way that this was developed. Turns out photosystem 2, even though it happened second, was discovered first. I'm sorry, I botched that. Photosystem 2 happens first. It was discovered second. That's why it's called Photosystem 2. Photosystem 1, even though it happened second, it was actually discovered first. So they're named rather in the order of their discovery, not in the order in which they happen or take place. All right. So in Photosystem 2, I'm going to go to a new slide here. In Photosystem 2, we have a special pair of chlorophyll molecules, again, the ones that donate their electrons, and those are called P680 or P680 because they have a peak absorbance at a wavelength of 680 nanometers. So we call them P680, okay? Photosystem 1, which happens second in the process, has a special pair of chlorophyll molecules we'll see in a minute, and that's called P700 because they have a peak absorbance at 700 nanometers. Okay. All right. So what we're talking about here is two photosystems that electrons get funneled through, and in both they pick up energy and become excited. Between those two photosystems, there is an electron transport chain, basically the same thing we saw in Chapter 9. And what do electron transport chains do? They extract energy from electrons and use that to build ATP. And that is exactly what's going to happen here, okay? These are going to be extracting energy that's used to build ATP. All right, why? Well, we need ATP to run the Calvin cycle, which is the second part of photosynthesis that actually builds the sugar molecule. Okay, so let's look and see where we are here. This is a broad overview then again of photosynthesis. I showed you this earlier. So we are now in the light reactions. That's where our photosystems are. Okay, water comes in, provides the electrons. Oxygen goes off into the environment. Light comes in, excites those electrons, and bumps them up to a high energy state. That's used to make ATP, which then powers the Calvin cycle. We also get electrons loaded into NADP+, reducing it to NADPH. That provides electrons and hydrogen as building material to combine with CO2 to get our sugar. Okay. All right. So here we have the light reactions. Okay. This is the light reactions. On the left we have photosystem 2. Again, it happens first. On the right we have photosystem 1. Happens second. Okay. So we have water coming in, water being split, the oxygen going off to the environment. Electrons get funneled into P680, okay? Light comes in, a photon of light, bounces around the chlorophyll molecules like I talked about. Ultimately, its energy is donated to P680, and it takes those electrons and bumps them up to high energy, primary acceptor. Primary acceptor grabs onto them and prevents them from falling back down and giving off their energy. All right, then those electrons move down an electron transport chain. It even has a cytochrome complex proteins like we talked about in cell respiration, the transport chains there. And so there's going to be a proton gradient produced here by the energy given off by the electrons in the same way it was in respiration, and that's going to be used to make a lot of ATP. All right, electrons move down the proteins of the chain, giving off their energy. That's used to make ATP. They are then donated at the bottom of the chain to P700. P700 then, with another photon of light whose energy comes in, bounces around, and eventually goes to P700, bumps those electrons up to a primary acceptor a second time, and this time they're passed down a short transport chain whose point is not to release their energy to make ATP. The point of this transport chain is to load those electrons and the protons following them into NADP+, reducing it to NADPH. The enzyme that does that is called NADP+ reductase. All right. So now we have two energy products. We have NADPH providing electrons and hydrogen for the Calvin cycle, and we have ATP providing the fuel to run the Calvin cycle. All right, so this is sort of a cartoon analogy for this. Photosystem 1 on the left, photosystem 2 on the right. Photon of light comes in, bumps an electron up to high energy. Primary acceptor grabs onto it, prevents it from falling down and losing its energy, instead passing it down a transport chain where the energy is siphoned off slowly and used to build a proton gradient that's used to make ATP. Electron comes off the chain, goes to photosystem 1, P700, bumped up again, and this time primary acceptor grabs it and passes it down a short chain, which loads it into NADP+, thereby reducing it to NADPH. All right. So, what now happens with, or how is it that the energy of the electrons is used to build that ATP? Well, this is a process, again, called chemiosmosis. Okay, we saw chemiosmosis in chapter 9, cell respiration. That was where the proton gradient that was built by the energy of the electrons causes the protons to flow back through the mitochondrial membrane through the enzyme ATP synthase, and that makes ATP. That's chemiosmosis. We see exactly the same thing here. Okay, this is the same way to make ATP. The spatial arrangement of how it happens, how protons flow in the chloroplast versus the mitochondria is a little bit different, but essentially it's the same process. So let's take a look at what's going on here. First of all, in mitochondria, protons are pumped to the intermembrane space and drive ATP synthesis as they diffuse back into the mitochondrial matrix. So they're moving from the outside of the outer margins, I should say, of the mitochondria into the matrix as ATP is built. In chloroplasts, protons are pumped into the thylakoid space and drive ATP synthesis as they diffuse back out into the stroma. So they're flowing from deep inside towards the margins of the chloroplast. The direction of proton flow is what's different here. Okay, so let's take a look. All right, on the left, we have a mitochondrion. Okay, so look at your color coding. The dark gray is the intermembrane space. That's the high proton concentration. That's this space right there. That's where you have water behind the dam, protons behind the membrane. All right, so protons flow back down their gradient through ATP synthase into the matrix. Low proton concentration, they're flowing from here into there. All right. In the chloroplast on the right, okay, what we have is the reverse. So the thylakoid space, that's the space deep inside the thylakoids, dark gray, that's where you have your high proton concentration. Protons flow from there across ATP synthase into the stroma. So they're flowing from inside to out. So the direction of proton flow is exactly the opposite. But it's still chemiosmosis, it's still producing ATP using an ATP synthase enzyme. Okay, it's again that water behind the dam analogy that we talked about. All right, so ATP and NADPH are produced on the side facing the stroma where the Calvin cycle will take place. The Calvin cycle is going to take place in the fluid here called the stroma. We'll see that probably in the next video. I think I'm going to make a part 3 on this one. So, in summary, light reactions generate ATP, and they increase the potential energy of electrons by moving them from water to NADPH. All right, so here's our summary, and I think this is going to be the last slide for this video because I'm right at about a half hour. Okay, what we have is a single thylakoid, right? That's one plate or disc, okay? And we have the thylakoid space where we have a high proton concentration. We have the stroma out here where we have low proton concentration. Here we have photosystem 2, there's photosystem 1. Light comes in, water is split, providing electrons that resupply P680 with electrons. Electrons get bumped up, they move down the transport chain. Their energy is used by active transport pumps here in the cytochrome complex to pump protons from low concentration in the stroma to high concentration in the matrix, or I'm sorry, the thylakoid space. Okay, we'll get back to that in a minute. Electrons have now moved down the bottom of the chain. They get donated back to P700 of photosystem 1. Second photon of light comes in, bounces around, bumps them up a second time to another primary acceptor, which passes them down a shorter chain, and where those electrons go, protons go. So electrons and protons are added to NADP plus by NADP plus reductase, thereby reducing it to NADPH. And that's going to go over to the Calvin cycle, providing hydrogen as building material and high energy electrons that become energy content of our food, sugar. All right, now let's go back to our high proton concentration in the thylakoid space. Protons don't like being in high concentration here and low there. They can't get through the membrane, they're ions, so they drop through ATP synthase. Again, that's exergonic flow, right? It's like water flowing through a dam. It gives off energy, and the ATP synthase molecule uses that to expose active sites that accommodate or fit ADP plus phosphate, thereby forming a bond between them, making ATP, high energy ATP, which then is used as fuel to run the Calvin cycle. All right, I'm going to stop there. That's going to be the end of part 2, and I'll have a little bit left to go with the Calvin cycle for a part 3 video that'll follow this one. Okay.